Hi, everybody. It's a pleasure to join you all for FD Day and to celebrate the research accomplishments we as a community have made this past year. My name is Frances Lefcourt, and together with my esteemed Scientific Advisory Board co-director and friend, Dr. Adrienne Gilbert, and with the guidance and support of our Scientific Advisory Board and FD Foundation President, Dr. Faye Ginsburg, FD research has had an exceptionally productive year. We were devastated by Adrian's medical emergency, but so thrilled that he's on the mend, and we wish him a complete and speedy recovery. As Dr. Kaufman mentioned earlier, this year was transformational for FD research. The first FD patient has received a state-of-the-art genetic therapy at the NYU FD Center via an invaluable collaboration with Dr. Adrian Craner at Cold Spring Harbor Labs and his colleagues at NLORM. In addition to that exciting major development is the considerable progress our researchers have made this year in both unlocking the mechanisms causing FD and in developing therapeutics that could significantly improve patient health. Moreover, we have sown the seeds for the next generation of FD scientists to ensure FD research continues into the future, with new investigators joining our community who bring fresh ideas and innovative approaches for developing therapeutics. Because of time limitations, today we can only hear from a small subset of these scientists, but please know that many other creative and dedicated scientists are at work writing grants to support FD research and developing new therapeutic approaches for FD, both in the United States and in Israel. So our first speaker will be Dr. Anil Chakuri, who was a postdoc with Dr. Sue Slagenhut at MGH, before starting his own lab recently at the Mass Eye and Ear Institute at Harvard. He'll be giving you an update on the gene therapy program we have initiated to prevent the gradual vision loss in FD. Anil.
Hello everyone. Thank you all for attending the FD Day. My name is Anil Chakuri. I'm one of the investigators at Scapen's Eye Research Institute of Mass Eye and Ear Infirmary. My lab is majorly focused on developing a gene therapy approach to rescue vision loss in FD. As we all aware of, patients with FD exhibit loss of vision along with other neurodevelopmental abnormalities. The loss of vision actually starts from the first decade of life and and the patients progressively lose their vision, leading to severe visual impairment in later stages of life. Previous work from Dr. Lefcourt and others in the field has shown that the loss of vision is mainly due to the death of specialized cells in the retina called as retinal ganglion cells. My research is mainly focused on developing a gene therapy approach to rescue loss of RGCs in FD. With recent advances in retinal gene therapy and approval of several gene therapy products, including Luxturna, for human use to treat Leber's congenital amaurosis, gene therapy is the most expedited path for approval of new drugs and clinical translation. Retinal ganglion cells in FD retina are prone to cell death due to the loss of ELP1 gene in FD. Therefore, our goal is to deliver full-length human ELP1 gene to the retina mediated by adeno-associated viral vectors, which are commonly called as AAV. AAV is a non-replicative and safe viral vector that can deliver genes to the target tissue. Here in this case it is retina, which is a thin layer of tissue containing light-sensitive cells at the back of the eye. Dr. Lefcourt's group has generated a novel AAV vector that can express full-length ELP1 gene, which can be targeted to the retinal tissue. Concept for retina-specific gene therapy is from Dr. Lefcourt's previously published work indicating that AAV mediated delivery of ELP1 vector to the retina intravitreally rescues retinal ganglion cell loss in FD mice. Here in this graph, we can see the rescue of retinal ganglion cell loss in the retinas injected with AAV2 human ELP1 vector. um, compared to the untreated mice, which indicates that delivery of AAV2 ELP1 vector can rescue retinal ganglion cell loss in FD. Continuing Dr. Lefcourt's work, we have started testing the visual function defects in conditional knockout FD mouse model. Our results indicate that thinning of retinal nerve fiber layer, which was measured by OCT, pattern electroretinographic responses were reduced in FD mice compared to the control mice at 3 months of age. In addition to that, visual acuity was also measured in FD mice, which indicated significant reduction in optomotor responses in FD mice compared to the control mice. Therefore, we plan to use this mouse model to further test the efficacy of AAV2-ELP1 vector. There are 2 ongoing studies in my lab currently. The first one is a dose escalation study to test the effective dose range of AAV2-ELP1 vector. Basically, we will inject different doses of AAV2-ELP1 vector in the eyes of FD mice to determine the effective dose to be used in FD patients. In addition, we are also performing a pilot study to test the safety of AAV2-ELP1 vector when delivered intravitreally to the eye. We will mainly focus on testing immunological responses caused by AAV2-ELP1. Results from both these studies are critical in moving the gene therapy approach to the clinic. In future, we will focus on detailed safety and vector biodistribution studies in mice and also potentially non-human primates to obtain the necessary preclinical data that is required for IND approval of the drug. We are collaborating with Tycoon to acquire the necessary funding for performing these preclinical studies, which are essential for IND approval. With that, I would like to thank FD Foundation for giving me the opportunity to present my work in this meeting. And I would also like to thank Dr. Sue Slogan-Happ from Mass General Research Hospital, who actually introduced me to the world of FD and mentoring me. I would also like to thank the members of Sue's lab for their continued support, which is critical for performing this research. Special thanks to Dr. Francis Lefcourt for graciously providing all the tools, the research tools important for performing this research and mentoring me. I would also like to thank Luke Vandenberg for his mentorship as well. A big thanks to funding agencies, National Eye Institute, NINDS, FD Foundation, and Knight Templar Psi Foundation. Thank you all.
Thank you so much, Anil, for that great update. And we'll be talking more about gene therapy if people have questions in the Q&A session after this session ends. Okay, so next, it's me, and I welcome to my generic conference room in a hotel in Western Pennsylvania where my husband and I have traveled for a wedding. So I'm just going to give a quick summary of a brand new paper that we just published in collaboration with the NYU Center, with Dr. Kaufman, Dr. Gonzalez Duarte, and the ophthalmologist, Dr. Carlos Mendoza Santi Esteban, on the retina in FD. So in both the FD retina and in a mouse model for FD, together what we found that's novel, and with a group in Spain, University of Alicante, Dr. Nicolas Cuenca, what we found is that in addition to the loss of retinal ganglion cells, there's also an inflammatory process that's at play in the retina. And that's marked by an activation and overgrowth of another cell type in the retina that are called glial cells. And actually this glial response actually precedes in the mice the degeneration of neurons. So we've all known that for several years that people with FD progressively lose their vision due to the slow degeneration of the neurons in the retina, in particular one particular kind of neuron in the retina, the retinal ganglion cells. But what we've shown is that in addition to the loss of neurons, not only now do we show that glial cells are impacted, but we also found significant changes in the vasculature or the blood vessels. And this is important because in the retina, the retinal ganglion cells and the glial cells and the vasculature all form what's called a neurovascular unit. And those three cell types communicate extensively. So if your neurovascular unit, meaning the glial cells and the vasculature, are not healthy or activated in some or inflamed, then the neurons are not going to be healthy. And so the good news is that this provides additional targets for therapeutics because other optic neuropathies also have problems with vasculature or glial cells. So now we can start to take advantage of that literature to try to reduce the glial and vasculature responses in the retina in FD. So this work was headed by my PhD student, Dr. Anastasia Schultz, and could never have happened without the support of a very special FD family and the interdisciplinary team of physicians and scientists at the NYU Center, University of Miami, and at the University of Alicante in Spain. Okay, and then finally, I'm honored to introduce our last speaker in this session, Dr. Sulu Sagenhub from MGH, who you all know well as the discoverer of the ELP1 gene mutation in FD and the developer of small splicing molecules, which she and her team have demonstrated are extremely effective at raising ELP1 levels, which, as we all know now, is the protein reduced in FD. So I turn it over to Sue.
Thanks so much, Frances. Let me share my screen. It's really nice to be here today and to see all of you online. As you heard, my name is Sue Slagenhop, and I am at Mass General. And I just want to make sure you can all see my screen. Looks like you can. So I'm at Mass General Hospital, and I'm really speaking on behalf of not only myself, but also my team. I'm a professor of neurology at Harvard Medical School, and my lab is in the Center for Genomic Medicine. And I've been working on FD for over 30 years now. I started working on FD when I was a postdoctoral fellow, and I'm really thrilled to be here today and to tell you a little bit about our progress of late. Now, the title of my talk is Developing a Drug to Treat FD. And you heard the really amazing news already that this year, for the first time, an FD patient has received a genetically targeted treatment. And so you might, and then you heard Anil talk about the development of gene therapy, and you'll listen to me talk about developing a drug to treat FD. And you might ask yourself, okay, well, we have a therapy. Why do we need other therapies? Well, our goal, and the goal of all of the investigators working on FD, is to not stop, but to continue to work to identify multiple treatments. You saw what's involved in getting an ASO gene therapy. That might not be the best route for treatment for all FD patients. And so the goal of my team that I'll tell you a little bit about today is to develop a drug. So we've been working on this for quite some time. Many of you have heard us talk about this before. And earlier, Dr. Kaufman gave a beautiful explanation of what goes wrong in FD. What I'll simply say here is that in this splicing mutation in the ELP1 gene, where we join exon 19 to exon 20 to exon 21, and this mutation here causes this to be disrupted. And so in FD, in our cells, sometimes we skip exon 20. And what we want is to improve the inclusion of exon 20. So our quest has been to develop a drug that would somehow interfere with this process, somehow make this process better by binding here at the end of exon 20 and leading to the production of more functional ELP1 protein. And our goal by doing this is to stop this from going this way and to make it go this way and lead to ever increasing amounts of ELP1 protein in FD patients. So how have we been doing this? For many years, we worked with funding from the National Institutes of Health Blueprint Neurotherapeutics Network. We had our first grant in 2012, and we worked for several years on a drug. We then worked with a company called PTC Therapeutics, who have been really remarkable collaborators. Unfortunately, in 2019, they decided to discontinue the clinical development of the drug. So we took it back with their support and have continued to work on it and received a second grant from the National Institutes of Health. We work very closely, as you can see from this team, my team here, with Dr. Kaufman and Dr. Gonzalez Duarte at the NYU's Dysautonomia Center. They're a critical part of our team. And then we have many other specialists that are involved in drug development from both private industry consultants as well as the NIH helping us. And our goal here is to bring all these experts together to develop this drug to treat FD. So what have we been able to do? We got the grant again last year and started our studies in earnest. And I've defined these terms for you that you might hear a lot when we talk about drug development. Pharmacology is the study of the effect of drugs on animals and people. So we've made significant advances in understanding how our drug increases ELP1 protein, and we've shown that it rescues the disease in FD mice. Toxicology, which is the study of the adverse effect of drugs on animals. We're working on studies to test what the toxicology looks like for our drug. And to date, we see that the data supports continuing to develop this drug. DMPK is an acronym for drug metabolism and pharmacokinetics, which basically is the study of how drugs, when we take them, are transformed, broken down, and cleared in the body. And so again, so far our studies suggest that the drug should be tolerated in FD. And so work is underway to determine what dose would be effective. And then chemistry. We have made a lot of progress in learning how to manufacture the drug. The original drug that was made by PTC Therapeutics was manufactured in China. Now we have a United States company, MRI Global, because this is one of the criteria when you're working with the National Institutes of Health, is that the work is done at US companies. And so they have successfully learned how to synthesize the drug. So we're currently working on studies to determine how much drug can be tolerated in animals. MRI Global is about to begin synthesizing a very large quantity of the drug so we can do what we call IND-enabling toxicology studies. These are the final toxicology studies to prove that the drug is safe enough to go into people. So we've already started talking to the FDA about our plans for the toxicology studies and the first-in-human studies. We've worked with Tycoon and Adam Sachs and Francis Lefcourt and others to submit applications for PTC 680 to be designated an orphan drug and for FD to be designated a rare pediatric disease by the FDA. And we are, of course, working with Drs. Kaufman and Gonzalez Duarte at the FD Center to begin to think about how we might do clinical trials for a new drug that would be an oral drug or a drug that could be taken through the gastrostomy tube. And then just one other note in other news, we also received a new NIH grant to work on FD that's entitled Understanding Neuronal Cell Type Specific Dysregulation in Familial Dysautonomia. Our goal here is to really understand which cell types and these cell type specific gene networks are disrupted when FD patients lose ELP1. So we will use very advanced tools and patient-derived neurons, and we're also working with Nadia in Georgia to work on this study, and she's a collaborator with us. And so we want to determine why specific neuronal cells die in FD. So this will hopefully, when we have a better understanding of what cells are lost or what's going wrong in FD, will lead to, as you heard Francis just talk about, other routes to therapies. So if we can't attack FD by attacking the gene alone, we'll find other ways to treat FD. So I really want to thank my team shown here in this picture. I especially want to call out Dr. Elisabetta Morini, who is an assistant professor in the group and who really helps me by driving the studies forward. And all of these members are integral members of the team, and really we wouldn't be able to move this drug forward without all of their help. And I really want to thank all of them. So thanks.
Thanks so much, Sue. That's really exciting, and congrats on your new grant too. That's on top of what the Blueprint program. That's wonderful.